honors by graduate, Shri Durga Prasad Ji is a distinguished coordinator of Honeybee AP project based in Hyderabad. To his credit, wherein innovative technologies and products are identified by dietary and nutritional factor in the prevention, treatment of secondary complications of diabetes. Guest for today evening, Dr. Jairam Valadi. Please. Dr. Jairaman is an AP Bionet India representative and currently serving as a scientist at CDAC Pune, the laboratory Pune. His research interests are in lieu of applications of artificial intelligence and machine learning. Supriya is a project associated at NIN, and what more? Congratulations to both of you and scientists for their outstanding contribution in the area of functional genomics and bioinformatics. 2. Genders, Megamotive Database, DD Database, SWAP, and iMotive DB. And also web servers iMot, Harmony, ScanMot, Dial, and SMotive. Each of these works are published in international journals. These involve setting up computational tools for the automatic recognition of structural and interacting motifs in proteins, curated knowledge base of domains involving in swapping. Most prominent of the databases is the genders, where more than 4,000 structural domains are connected with roughly 10,000 genomes of model organisms, and the connections are stored according to structural and taxonomical hierarchies. Genders is one of the highly accessed websites from the laboratory and has 5 lakhs visits from external IP addresses. Besides working on databases and web servers, he has contributed in areas in prediction by machine learning approaches like support vector machines, SVMs, for identification of ordnance binding proteins in whole genomes, SVM and random forest method for prediction of domain swapping of proteins from mere sequence information alone. Pugalanti has 34 publications, out of which 21 papers have emerged from Soudamani's lab alone, and several of his papers are highly accessed. He has the uncanny ability to deal effectively with large amounts of data. He also brings collaboration between Dr. Soudamani's, Sugannathan's, and Kolatkar's lab. Pugalanti, he is a soft-spoken and modest individual. He comes from a very simple family, and we believe recognition of his contributions to efforts. It is a great honor for Bioclues to present him with Bird Award 2011. Unfortunately, Pugal couldn't make it today. May we request Shri Durga Prasad Ji to present this plaque, honorarium and certificate to him, please.
Good evening to you all. The chief guest on the dais, dear friends. Actually, today morning only Dr. Ganeshan has called me and asked me to collect this award. I don't know him by face because one of my friends is working there at KAUST, the King Abdullah University for Science and Technology, and those two were good friends there. So he didn't have anybody in Hyderabad to collect this award, so he requested me. So that's how I'm here. And honestly, just now, I've seen his picture on the slide. I don't know his face either, but I'm sure, the way he was talking and the way he was expressing, that really shows that he's a very good guy. And this now she has introduced that he has published close to 34 papers and he has done a lot of work on bioinformatics, and he rightly deserves this award. And I think the bird has, Bioclues has chosen a right person. I really appreciate Bioclues and bird. And I also congratulate Dr. Ganeshan for receiving this award. Thank you all. Thank you.
Can someone dream of a Nature publication as the first career publication? Yes, we do, and will innovative projects dealing with genome analysis from crop improvement program. He has made outstanding contribution in decoding mark study for next generation sequencing based pipeline. For instance, as an active member of the PGNP genome sequence, establish a benchmark by comparing different tools for NGS-based pipeline work is current in progress. Genome analysis for variety of applications and abiotic stresses, RNA sequence. In summary, he has contributed.
Because I'm feeling very happy. I'm not able to concentrate. But of course, I'm very, very thankful. I got this special mention. So I'm giving a special thanks to all Bioclues team. And I'm really congratulate also Dr. Pugal for getting award, bird award for this year. And I'm really obliged of you all people who gave me such a precious award. Very, very thankful to you all. Okay.
New platform for me to address, because I am more familiar with people with high knowledge but low education. We work for, this is an organization called National Innovation Foundation, and every state we have non-virtual organization where we coordinate with the grassroots innovator to this national body. We all work as volunteers, and it is in almost in all the states. So here it is called Honeybee AP, like in South, Honeybee Tamil Nadu, TN, Kerala, Karnataka, then UP, MP and all. In our country at the moment we have around 1,80,000 innovations innovated by the grassroots innovators. We call grassroots innovators who studied under 10th class. And most of our innovations are need-based. So, totally uneducated, some educated to the basic level, some daily wage earners. So, we find all these things in the rural area, mostly in the rural areas and the hilly areas. So, we travel a lot. We identify the people, which is more a difficult thing. So, but it's past 10 years, so we have on the traditional medicines, herbal, and engineering, medical, everything. So usually when we interact, many people will wonder why, how these people can innovate. But we feel proud and happy that in our ancient Indian knowledge-based system has lot of forgotten knowledge. So we try to get that forgotten knowledge. So we travel to the rural areas called Shodh Yatra. We travel for 10 days in the most rural, hilly areas of the country. Once in 6 months, around 100 to 120 people will come and join. Few people come from abroad to do that. It is a non-pleasure walk for many people because they do not have any basic amenities. The only thing, all the expenditure has to be met by all the people who participate in that. Only thing is we provide the meal, but we charge to their account. So it is because food cannot be given at most of the places. So we have our own cooks and all, and sometimes some villagers give us the food. So like the basic amenities like toilets and all will not be there, no cell phones because most of the areas do not have power. But when we go through those places, it is amazing to see the knowledge, which every 6 months it is completely new and amazing that we have got so much of knowledge. And last year it was at Jharkhand. In Jharkhand, we found out a person who heals all the bone fractures. Natural bone fracture, he says it is very easy. But even if there is a multiple fracture with all the broken pieces and all, he sets it with massaging only. And he ties a 3 inches bandage to his shoulder or to the leg, wherever there is a fracture. And immediately after that, he applies some herbal thing. Then he asks them to lift.
Last September, it's always a great honor, to talk to him, to take his suggestions, and also, to see that, the way, he motivates, the youngsters. And I'm sure, that during coffee time, during tea time, I think you'll have, more networking, with elite people, on the dais. And at this juncture, in fact, we are very much, privileged to recognize, certain people who have been within Bioclues, for the past few years. So, may I recognize Safia Fatima, who has been here. I'm sorry, I cannot see people somewhere there. If Safia Fatima is there, if she could stand up. Tormenting her, so she had to leave. We recognize the presence of Mrs. Safia Fatima. She was in US and she was the foremost member of Bioclues way back in 2005, ever since when we started it. We recognize the presence of Murli Subramaniam Jandiala, who is the founder of bharatujog.com. If you could stand up, Murli, please. And if you can take one small rose from us, yeah. We Sarwar Azam. We also, please.
Yeah, we have Prashant actually asked Dr. Shashikaran to come to this function, but because of his other commitments, he could not come. Make some comments on what Prashant asked me to. I met Prashant sometime, I think about 4 or 5 years ago in Pune, in a workshop in CDAC, and since then we are in touch. But really, Bioclues is a virtual organization and volunteer organization, and one can do this kind of wonders. It actually surprises me. But that apart, he has been tiring a lot for the last couple of months to organize this event, and what else we could support than just providing a hall? That's all. And I just convinced my director, and he was generous enough to say that, okay, go ahead and have this function here. That's it. Nothing more than that we deserve. But then, what actually Bioclues is doing is a great job as a volunteer and organization, encouraging the young people, particularly in the area of bioinformatics. When I say bioinformatics, I'm not a bioinformatic related person. Basically, I'm an experimental biologist. But I do involve some of the bioinformatic tools, not very extensively, but wherever and whenever it's necessary. That's how I used to attend some meetings of bioinformatics, like one I attended at CDAC and here and there. But what I see, like if there are any bioinformatic people here who would like to make their career, there's a lot of scope in this area, particularly from the point of Indian context, like what elite person Durga Prasad was saying, that just don't take that what is going on as a trend. Like everybody does some kind of typical bioinformatics. Now, if I'm not criticized for this, bioinformatics means many of the guys think that just aligning some sequences and putting some homology modeling, that's fine. But I think you have more than that to do in bioinformatics. I'll just take some example, taking the clue from Mr. Durga Prasad, that I have been actually talking to Prashant. If any one of you are serious in making a dent in the area of bioinformatics, putting our information into that, is pick up all the knowledge that is there in our traditional system, be it Ayurveda or be it in our traditional nutrition. What are that we have, what are their molecular functions, how they work, create the networks, and provide the information to the other guys. I'm only touching on the surface, I'm not going to the details, but if any one of you are serious, we can sit across the table, come up with at least a couple of projects or a few projects, which I have some ideas, but I'm not a bioinformatic person. So what you do is, you create the database. What are the molecules that we have in our tradition system? take like what no, Durga Prasad was saying, that which cures such a disease. Like that, we have hundreds and thousands of information and molecules with us, and then provide their molecular information. Most of it is available, but put it in one umbrella, or you put it in one database, and then give their molecular action, create networks, how they work, and then you also provide on the information on the nutrients. Now these days, what we always used to think vitamins or nutrients, like say if you take vitamin A, it only functions for vision, but vitamin A does one other functions. Can you put this information, how it works, and but it doesn't do in isolation. There are other circuits and networks that are connected with it. If you disturb one circuit, entire thing get disturbed. Can you provide that from the bioinformatic point of view? Then challenge the experimental guys to prove it, or you work with them, or you, if you happen to be an experimental guy, you yourself prove that by experimentally also. So these are a couple of things I just wanted to share with the bioinformatics guys, if anybody who is really interested in taking up the projects on bioinformatics. And with my limited knowledge, I can always provide some inputs. Then coming to the point that I'm a little bit touched by the words what Mr. Prasad said, Durga Prasad, innovation doesn't come from glass buildings. Innovation doesn't come from education, like higher qualifications, PhD does not mean that we are innovative. On that front, actually what we are doing, I work for an NGO. I spend a day or two in a month. The foundation name is called Vande Mataram Foundation. But we don't focus on innovation. What we focus is that we go to rural areas. We focus on rural government schools. We think that education or all that knowledge doesn't only exist in metro cities or in glass buildings. We think that knowledge there in the remote corners of the country. But then we need to facilitate that. We need to give some push to that. So what we do, we have actually about 200 plus and 200, almost 500 schools in our fold in 2 districts. We reach to almost these schools, government schools. We try to improve their education standards. Let's say if the school curriculum or the teachers are not taking care of it, we counsel them, actually we inspire them, and then during the 10th class time, actually we pick those guys and we put them in a camp. In 1 month, we cover the entire 10th class syllabus in 1 month, and in the first batch, it's 5 years now, in the very first batch, from 50 students, we started with 50 students, 16 of them got into IIIT. 16 of them got into IIIT. So what I'm trying to say that innovation, knowledge does not exist only in metro cities in glass buildings. It is there all over. We, people who have come, because I have some obligation, because I have come all the way from a small village, and I feel it's my responsibility to do something back to the society. That's why I'm involved there. But it's not that I do a lot of work. There are other guys who spend, actually a couple of people who spend 24 hours a day, but I spend a day or two in a month. So this is what we are trying to do. So now there are these guys who are in the IIITs are three places in Andhra Pradesh, and some of them are in engineering, some of them are in medicine. We provide, if they are poor guys, we provide the books, we provide the school fees, we provide the transport. But then they want to come to the stage what they want to do, and then later they have to go back and do the same thing what we are expecting. That's what I think most of us have to do. So leaving it at that point, just as Prashant always tells that I'm a young scientist, well, now I'm not anymore because I crossed 40. So what actually this young scientist or recognition means to me is in a different way. Actually, they don't really affect my day-to-day this thing, but I think many people tell me that sharing this with others will inspire them, because I have come from a small government school in a remote village, and facing all the hurdles on top of that any other person faces, a physical disabled person can face. And at every stage, at some point of time I have decided what I have to be. Then to come here, I think it requires a lot of determination. But then now there are so many organizations like this which can help the people who have to reach to their destination. That's what we need to do. But as far as the bioinformatics and Bioclues are concerned, we have a lot of scope doing the work from the point of Indian context and from traditional information, or from the nutrition point of view, for that matter, even other molecular aspects, what people are sitting here from CCMB and other places. We can take their help and make a dent. Don't do just for the sake of doing a bioinformatics, getting a certificate and claiming that I have a certificate in bioinformatics or I have a diploma here. but make a difference. That's what is required in the present stage. And of course, it's a competitive world, then only you survive. So I think Bioclues will help you to do that. That's what I understand from Bioclues, what Bioclues is doing. So you take their help and try to reach your goals. Thank you very much, Prashant, for giving this platform. Thank you.
All of us. The reason is over 89 colleges are removing, I've already removed the taught program in bioinformatics the last academic year. And if you see the next academic year from 2012 to 2013, there are over 159 colleges where bioinformatics taught program is removed or is to be removed. That's really, a very alarming story. The reason is there's lack of competent faculty. That's because either people do not know what bioinformatics is. That's just because bioinformatics is a tool. They only venture into bioinformatics just because they consider it as a discipline. So that's the reason why we, of the several virtual workshops that we kept on organizing the last 6 years, we also started on a mission called Mission Vision 2020, wherein we thought of setting up a vision wherein we wanted to educate 2020 biologists, not other cross-disciplinarians, biologists, to take a shape into something called shape themselves into bioinformaticists, because they are the real people who can really nurture talents. That is our main motto. And thank you, Dr. Banu. Will definitely reach unto your expectations. Before we come back to Shri Durga Prasad Garu again, we recognize the presence of Ms. Maithili, who passed out her MSc from UK. If you can stand up, Maithili, please. You're just, behind this. You'll get a quick rose from our Bioclues team. Thank you, Ima. We also recognize the presence of Rajesh of Wellness Care again. Rajesh, if you can stand up, please. Yeah. Those three gentlemen who are sitting there, N.V. Rao, Rajesh and Ramulu, you can just ask them, in your welcome kit, there is a wellness brochure. Only first 25 participants are the luckiest ones. I think, you'll get a discount of your wellness clinic things or hygienic things initiative. You can really, discuss with them, during a quick cup of tea. After Dr. Banu's speech, Durga Prasad Garu came up and he just wanted to talk for 3 to 4 minutes. Please, yeah.
By name Dr. P.D.K. Rao. I think he came back from Chicago. He got a nuclear physicist. He was a nuclear physicist. And after that, he thought, okay, let me go back to my own state and work. Somehow he went to Visakhapatnam and he asked the district collector, I want to work in a rural area. So he said, okay, what is your background? I'm a nuclear physicist and went to US some 30 years back. So he said, what is that you are going to do? I'm not talking about nuclear physics and all, but let me go by a village. You just suggest me one place. So he suggested one place called Chipurpalli. He went there. Then he started going through the problems. Because he returned back from US, he was termed as CAA man because he was protesting for all the illegal things. Then he thought it is not the right thing. He said, what is the origin? So there is no education. So he thought, okay, let me first start educating the people. And he started a system with which people can learn. And he confined that to Srikakulam and Visakhapatnam, Vijayanagaram, small pockets. It was recognized by late our great man P.V. Narasimha Rao Garu when he was Human Resources Minister. And he said, why don't you adopt this across the country? So he said, no, I don't. I can't do this across the country and I don't want to get involved with the government system. Then he was doing that in that area. And one Mr. Manohar Prasad, a retired, he took voluntary retirement from civil service man from AP cadre, went to Adilabad district in Utnur Mandal because he worked there as collector, joint collector and all. So they said, sir, my son is studying in 10th class, but he can't read a 6th class book. So why give them a good education? No money, sir. So I said, if you don't spend money on your child, why will I spend? If you are willing to spend money, then he said, we don't have money. He said, okay, you cut down 1 cup of coffee per day or tea per day. Give me that 5 rupees and we will see that your son gets education. They said, they agreed. And he said, you don't give me money. So all mothers in the village formed one organization called Mahila Samajya. So these organization first sent a girl or a boy who studied from 9th class to 12th class to this PDK Rao's place at Chipurpalli, got them educated, teachers training. So they teach by way of dancing and singing. Teacher
Prasad and Dr. Banu Prakash, I think, and I would like to say only a few words. As all we know, in the last few decades, biotechnology has made rapid strides, and with that, the bioinformatics also has grown up. Now, with the symbiotic relationship between biotechnology and bioinformatics, a lot of wonders can be done, especially in personalized medicine and drug-oriented research. Now, I would like to say that bioinformatics picks up domain data from biotechnology and life sciences, and then try to provide knowledge by using various tools. Now, these tools are taken from interdisciplinary areas, from different areas of science and engineering. Now, for an example, if we require to analyze gene expression profiles and to come up with whether a person is having a cancer or not, or to pick up the most informative features, we need to use tools from machine learning and from the most informative features and most informative genes which correlate to cancer. So to get such sort of knowledge, a bioinformatics person has to read and understand different fields of science and engineering. So he has to think in interdisciplinary way. So for that, I think he has to study a lot of material from different areas of science and engineering. If a person from village, as Professor Durga Prasad told, can pick up and then provide machines which is useful for the society, why not people from bioinformatics can learn for interdisciplinary areas, assimilate and then provide knowledge and then use it for the usefulness of the society. Now, people used to take life sciences as an option because of not of love of the life sciences, but because of fear of mathematics. But that scenario has now changed. People now, there used to be a saying now, a computational biologist in India will talk biology to computational people and about computer to biologists and biology to computer scientists. So that scenario has to change, and people have to be ambivalent and learn a lot so that they can serve the society, and then they can bring about good research which is useful for the country. I would like to say, people, the young bioinformatics people, young researchers here must try to work hard so that they can concentrate, and then they must take somebody as a role model, and some agenda, an agenda which is useful for the society and for the country. So I would like to say, with this, I would end my speech, and I would like to wish all the young people a good career in their life.
I the presence of Shri Ananta Garuji. Thank you, Prashant. Thank you, Hima.
Because this team gave me an opportunity to share my little knowledge to high-profile scientists like Dr. Jairam Waladi, Dr. Bhanu Prakash ji, Dr. Durga Prasad ji. So I'm really once again, I'm very much thankful to you all. So before I start my presentation, I would also like to give my own comment about this talent and innovations. As sir told, Durga Prasad sir told that we have to look for some innovation in agriculture research so that we can reach to poor farmer. Sir, it's our institute which has a mandate for this. They are working on 5 crops, and the main mandate is to work to do some innovation so that we can make resource poor farmer happy, and that's the main goal of our institute. But it's a UN institute, so the area is not limited to India, but the whole world, especially semi-arid tropic, and particularly Africa also. And again, Dr. Bhanu Prakash was saying that talent is not limited to only metros or cities. Even I agree with him. Even myself from a village where last year electricity came. So when last time I went to my home or my village, first time I saw electricity there, or fan, or bulb. So you can see that. But I think Government of India has done a lot of things to capture the talent at a local level, and I think I am also one of the product of that initiative of the Government of India. My schooling is from Jawahar Navodaya Vidyalaya, and that is a government program to capture the talent at very early age of the student, like in 6th class only. And they recruit in a boarding school, which is of course Jawahar Navodaya Vidyalaya in every district, and give full fellowship, lodging, fooding, everything free till 12th class. And I'm also one of the friend. And nowadays, if you will find it, whichever the post, you will find some of the Navodaian are there. And I'm very much sure there will be some Navodaian in this hall also. And I'm one of that. And for that, I'm of course obliged of Government of India. And I think this program was started by Rajiv Gandhi to capture the local talent and give the facility of Doon school or something like that. So I got a very less time to present this whole work. Only 15-minute time was said by Prashant. So I just highlighted. I think light is not clear from here. Can I move the dice ahead? Sure. Impact overall agriculture production in the world, and I have just shown the slide, and I think it's a PPT version, so animation is not coming anyway. So because of the climate change, the temperature is increasing, there is also increase in the water level and other things, and that's going to hamper the crop productivity. But it's not always. There are some factors which will help the production of crop or cereals. But in totality, or the way it's rising, if it rises much, it will definitely going to hamper the crop production. So in a FOA study, there is a much pressure for breeders and to release the variety which can produce more and more amount of the grains, because every year we are increasing almost 32 million ton, but worldwide. But requirement will be after 2007 44 million ton, and by 2050 we will be having a world population of 9.2 million, and for that we will be requiring a very high amount of cereals, and the growth, the kind of growth we are expecting, is not fulfilling that point, that level. In that case, our ICRISAT is working on this direction, and of course, our mandate crop is 5, 3 is legumes and 2 is cereals. So legume will be the one which will be
fulfilling the deficit of the cereals. Out of these five crops, two is cereals, one is sorghum, and another is pearl millet, and the rest of the three is a legume, which is chickpea, pigeon pea, and groundnut. There is a lot of potential in pigeon pea and chickpea and groundnut, I mean all the legumes, and we can increase the amount. If we increase the productivity, then we can certainly not only fulfill, but we can share the load of the cereals on the legumes. And for that, different departments are working on the different plant, and we are working on the pigeon pea. And pigeon pea is one of the protein-rich grain, which gives the balanced diet, then very balanced than cereals, and it has a lot of potential, but the actual potential is very less nowadays. I mean, potential means potential of productions. So it's a rain-fed crop, often cross-pollinated, and it has a 22 chromosome in the diploid stage, and its size is 833 MB. So if you see the production in the last 50 years, so productivity remains almost constant. That is around the 700 kg per hectare, and it is little more in the Asia, but less in the Africa. But production has increased between 70s to 90s, and this increase is all because increase in the land, agriculture land or cultivated land. It's not the increase in the productivity. And it was estimated that by some scientists, it is 2500 kg per hectare can be produced. That is the potential, but actually it's only producing 700. So there is a large gap between the actual and the potential, and we are now trying to fulfill this gap. For this, there are a lot of challenges, and there are some biotic challenges as well as abiotic challenges, because for Fusarium wilt and mosaic virus, all these are causing the biotic challenges, and there are drought conditions, salinity conditions. These are giving the abiotic challenges to grow this pigeon pea and increase the productivity. To overcome these challenges, we certainly require some SSR markers so that breeders can, or SNP markers, or breeders can produce a high yielding varieties, and that can be released into the country. But the problem before this genome sequencing was that very few SSR markers were known, and there were very few diversity in the core gene pool, and and map and other things. The other genetic resources was very less. It was not that much available that is found in other crop like maize and rice. So we cannot make a hybrid or make a variety that can give the very high quality variety, so that it can increase the production of the pigeon pea. For this purpose, we go one step ahead, and we sequence the whole genome of the pigeon pea, and that was published this November only in the Nature Biotechnology. It's the first grain legume, and it's the first orphan legume crop, and the second food legume crop after soybean, and totally it's the fourth legume, and the other two is lotus and medicago. So what kind of strategy we use in the sequencing is actually we made some library, and there were that we can define in the two ways. One was the small insert libraries, and other was the large insert libraries. Those which are more than 1 KB we kept in the large insert library, and those which are less than 1 KB were in small insert libraries. And there were totally 22 libraries were made, and we sequences those data using Illumina sequence, Illumina HiSeq 2000, and we totally made the data to 37.2 GB, and these all our the pair in reads, and again we went for the filtering of those data and removing the low quality reads and the low quality sequences or overlapping sequences or poly A and poly T sequences, and finally we reach 137 GB high quality sequences, which is 163.4x of the total genome. Then we went for the GC content analysis, and we found that like other genome, like glycine max, trichocarpa, and Ricinus communis, all are giving the same kind of distribution of the GC. And so it also showed that the purity of the data. Not only this, we also did the KMR analysis for the size estimation and the purity of the data, and we found that there is only single peak, and which says that the data was homozygous in condition. Then the overall strategy is already published in Zhu et al. In 2009. There, we took we fragment the genomic DNA, we sequence, and then we cluster the sequences, make consensus using De Bruijn graph method, and then finally we make contigs. So first reads were overlapping reads were joined together and make a small contigs, and then these small contigs were joined again to make scaffolds and using the large libraries. And when we have large scaffolds, then we again use back-end sequences or plasmids and marker data, whichever was available that time. It was only 833 marker data were available. So we use those data and make greater scaffold or larger scaffold or super scaffold. So finally we assembled this genome, and out of 833 we were able to assemble only 605 MB, and which is nothing, which is 72.7% or 8% of the whole genome. So that's only we could assemble, and out of this, around 70% we assembled with the greater confidence, where every scaffold is larger than 2 kb. We estimated the PGNP genome size 833, but the actual genome size, which will be estimated by C value only. So it may be high or low, and it may be little high or little low, and according to that, this percentage of the actual genome will fluctuate. So the total scaffold we made is 1,37,000, but out of that, more than 2 kb scaffolds were 6,949 only, and N50 were 185 kb, but after merging with the marker data and backend sequences, we reached to a level to 21.5 kb. And then we look for the characters, features in the genome, and the first feature we look for that was the transposon elements. And we find there are so many type of all the type of transposon that is present in other genomes, and there were 51.67% of the genome having that kind of repeat, and which is very much similar to other genomes like soybean, which has a 59% I think, and castor and grape has 41%. But there are other cereals like maize and sorghum which has very high percentage of T in the genome, like sorghum has 62% and maize has 85% of the genome. The total retrotransposon we found is 37.12%. Ideally, actually, transposon was divided into two classes. One was the DNA transposon, the other was the retrotransposon. So only few percent was DNA transposon, major was retrotransposon. Then we look for the gene space in the genome, and for that we used two methods. One was de novo, other was homology. In de novo, what happened? We took some tool which is very much specific to discover the gene within a genome or within a sequence, and for that most of the time they work on hidden Markov model, and for the to work it properly, we have to train those sequences, train those tools by giving some sequences. Of course, for de novo we use Osgaster, GNSCAN, and Glamour HMM, and we predicted many tools. The problem with these tools is that if you become little liberal in parameters, they will give so many false genes, and if you become a little strict, they will left so many genes. In that case, we also look for the homology based gene annotation, and for that we took the genes from Arabidopsis thaliana and other eudicot genomes like Vitis vinifera and papaya, and we simply map into our genome, and we found that there are good number of genes which are coming, and which is very much common. And then finally we make a redundant set using the Glean software that is called Glean algorithm, and we make a finally redundant set of 48680. When once we made, then we also try to validate that this gene space, that we have captured the whole gene or not, and we also try to validate the whole genome, so that the functional genome has been characterized or not, or captured or not, because we have only got the assembly of 72%, but this 72% is actually having almost 100% gene space or functional part of the genome. For this purpose, for the validation purpose, we had already transcriptome sequences from PGNP only for many genotypes and that were used for many mapping population for SNP analysis. And when we map back to these gene space and the genome, we found that more than 90% are already is mapping. It means that we have captured all the gene space, or whole gene space. The next step was okay. To validate also, there are some eukaryotic genes which is fixed, and for this purpose there are 458 genes which are known to be compulsory in every eukaryote. And we try to find that how many of that important eukaryotic genes are already in our genome. And for that, we use the CEG-MA core eukaryotic gene mapping analysis approach. And we found that out of 458, 453 genes were present in our assembly. It also showed that 98% coverage of gene space in our assembly. So rest of the genome sequence that we could not find or we could not assemble is actually the junk DNA. But I cannot say it obviously until unless we discover it, but it is very much probable. Then we compare this pigeon pea with the soybean, because soybean is the most closest relative, and we found that exons per gene is little less, but average exon length and other are very much comparable. And we also look for the centenary between pigeon pea and soybean. For this purpose, what we did, we looked the genes in the pigeon pea, and we tried to find the homology in the soybean genome. And we found that for every chromosome has at least one or two hit, at least two hit or more than more in the soybean gene space. And you will see that here, these are the chromosome of the pigeon pea, which are hitting the soybean chromosome. And if you take one, it will hit at least two soybean gene. So one block is actually common to the two block of the soybean gene. That again established that the soybean genome is duplicated after speciation of the pigeon pea. And we also go for the micro centenary level, and then we found that there are some micro rearrangement in the centenary, and these are the chromosome of this is the one chromosome of pigeon pea, and the 6 chromosome of the pigeon pea, and this is the first chromosome of the soybean. And we map our transcriptome or transcriptome to our chromosome. It means pigeon pea, and to the soybean, and we found that you see these lines. These are coming straight. It means they have shown the conservation of the centenary. But here there are some little rearrangement also. And these rearrangement, of course, has come after the speciation of the pigeon pea. When pigeon pea and soybean diverge, after that soybean went for the rearrangement. Then we also look for the genome duplication within pigeon pea, and we didn't found much like soybean, because soybean is a whole genome duplication. But in pigeon pea, it was only few. Only 28 block of the we found, out of which 3 were intrachromosomal, and the rest of them were interchromosomal. And those 3 intrachromosomal were on first one was on this one, 6, 2 intrachromosomal on 6, 2 block of duplication, and one was on this one, 11. And most fragmented chromosome was 11, which has having hit or homology with other chromosomes. Then we again find the orthology. We did some orthology analysis and tried to find that how the gene family is distributed among the legumes. And for that we took 4 legumes and one outgroup, that is grape. And we found that 4311 cluster of the genes common to all 5 eudicots, means it is common to all the genomes. And 903 cluster is very much specific to legumes. And there were some genes which is very much specific to pigeon pea. And these genes can be lineage specific genes. And for that we did the later analysis. And we found that there are 266 genes which can be established as an orphan gene or a lineage specific gene, which is only and only specific to the pigeon pea and not found in another genome. So summary of genome feature. Yeah. No. I told you here. We found that 15,000 gene is very much specific in these 5. But to find the orphan, we again did the analysis with the whole database wherever it exists, and then we only found the 266 orphan genes. And summary of pigeon pea genome. So we
organizing it, and we've been thinking of organizing it, for the past few days. What exactly they do, Alice calling us. The thing is, they spend. Have you ever seen, biologists not using mobile phones? Yes, there are people you will find. Have you ever seen, biologists working in the laboratory, listening to the music? Yes, you'll also find. And there are also biologists who work for 18 hours a day and who also spend during weekends doing, such social service. They give, tricycles to the crippled. They seek, some approbation and funds, from different people. And, they're all, before you. I think a big applause is needed. Thank you very much. I think, before we just catch over for tea, there's also a vote of thanks, a quick vote of thanks there. Perhaps I can just, invite Maheshwar. I think I also missed Maheshwar. I'm sorry. Maheshwar is also there. Perhaps, Himak, and also from other parts of the world. We have got 1,100 members, of which 400 are active members all over 30 countries. And we have got 12 virtual projects and 60 scientists have been involved. And one of the important, also keep these bioinformatics to systems biology workshops. We have virtual workshops taken care by these eminent scientists, and we also provide certificate and honorarium. And there's another workshop, and as I told you, it's coming up in Bangalore. It is at IISc Bangalore or even at Bruker Daltonic's office that Shiv is working in Bangalore. And we also keep organizing several
Hi, I'm Mona. I'm a member of Bioclues. Please enjoy Biocluing.
I'm Soumya. I'm a PhD scholar and I really enjoy being a part of Bioclues.
Hello, I'm Sri Manjari. I'm a PhD scholar and I've just joined Bioclues and hopefully be a part of Bioclues and enjoy it.
I'm Renuka and I'm a member of Bioclues and we have a Women in Biology section. We really enjoy being a part of it.
Hi, I'm Himakumari. I'm a doctoral student and I'm a member of the Bioclues and RCC, and I really enjoy being a system biologist. Thank you.
Hi, I'm Venkatesh, pursuing PhD, and I feel happy to be a part of RCC and Bioclues. Thank you.
Hi, I'm Srikant. I love to be in Bioclues.
Yeah, hi, good evening. This is Prash. I'm the founder of Bioclues organization. Bioclues works on 4 mandates and avenues, which is acronymed MORE, M-O-R-E, Mentoring, Outreach, Research and Entrepreneurship. Please join Bioclues organization. Please visit us at www.bioclues.org. We're also archived using Bioclues Google Groups, namely Bioclues at googlegroups.com. Keep Biocluing. All the best.
Okay, fine. Hi, I'm Shiv, Secretary and Treasurer of Bioclues. We are organizing several virtual projects. Why don't you join us? Same thing, repeat? Repeat? Yeah. Hi, I'm Shiv, Secretary and Treasurer of Bioclues. We are organizing several virtual projects. Why don't you join us? The Bioclues aims for science and society. Thank you.